Hello everyone, here's a video of Group 4 for LCL2 project. Our group explained the co-transport. First, to introduce the subject, let's start with a definition of cell membrane. The cell has a plasma membrane which allows exchanges between the inside and the outside of the cell. It is composed of proteins, lipids, and carbohydrates. Its characteristic is to have a selective permeability. In order to allow exchanges between the outside and the inside of the cell, there are different kinds of cellular transport. First, the passive transport with simple diffusion, osmosis, and facilitated diffusion. The second type of transport is the active transport, which is divided into two categories: the primary active transport and the secondary active transport, also called co-transport. On the other hand, there are cellular transport for molar exchange inside the cell, like endocytose. We are particularly interested in what is co-transport.
Co-transport, as known as secondary active transport, is a coupled movement of one molecule going down its concentration gradient with a second molecule going against its concentration gradient. The molecule which goes down its concentration gradient will let out some energy different from ATP. This energy will allow the other molecule to move up against its concentration gradient. The protein transferred used is a co-transporter. It's a carrier protein that allows the transfer of two different molecules from one side of the membrane to the other side. They require energy to change shape to carry the molecule through to the other side. Pumps with practice secondary active transport or co-transport are sometimes referred to as Cooper's carriers. These pumps use the downhill transport of one substance to drive the uphill transport of another. Cooper's carriers, like the sodium-glucose co-transport protein, do end up costing the cell energy because the cell must use ATP to maintain the sodium concentration gradient that this carrier uses at its energy source. But co-transporter does not use ATP directly.
Okay guys, moving on to the next interesting part of this topic, we have two types of co-transport which consist of symport and antiport. So what is symport? Symport is when two molecules are moving through the membrane protein in the same direction, while in antiport, it is when two molecules are moving in the membrane protein in the opposite direction. So guys, so what are the differences between symport and antiport? What can we see between them? What can differentiate between them? So firstly, in symport, it is actually a transmembrane protein molecule in cell membrane which transport two distinct molecules or ions in the same direction across the membrane. But in antiport, also same a transmembrane protein molecule in cell membrane which transports two distinct molecules or ions but in the opposite direction across the membrane. So, secondly, the difference between symport and antiport is, in symport, one molecule will move down along the electrochemical gradient, while the second type of molecule will move against the concentration gradient. But in antiport, we can see that one molecule will enter the cell, but at the same time, the other molecule will exit the cell as well.
So a symport is a co-transport where there are movement of two molecules in the same direction through the protein channel. So an example of symport co-transporter is the sodium-glucose co-transporter. It is present in the epithelial cell of small intestine, which is found on the apical membrane. It is the lumen part of the small intestine. So there are two types of sodium-glucose co-transporter. SGLT1 and SGLT2, they both work similarly. So as for the function of sodium glucose cotransporter, it is to maximize the absorption of glucose from the intestinal tract and also recover glucose from the proximal tubule of the kidney following glomerular filtration. So for the mechanism of sodium glucose cotransporter, 2 sodium ions and 1 glucose molecule are transported into the enterocyte cells. This uses the potential free energy stored in the sodium electrochemical gradient established by the sodium-potassium pump. There are low sodium ion concentrations inside the epithelial cell. As both sodium and glucose is entering into the cell, this is why we would need energy. The sodium influx from the lumen to the epithelial cell is coupled with glucose transport. So as we can see at this animation over here.
Thank you for the explanation from ZIHUI. Next, I will proceed to the second type of cotransport, which is antipod. Antipod is the movement of 2 different molecules in the opposite directions through a protein channel. And here is the example of antipod, which called sodium-calcium exchanger. Sodium-calcium exchanger normally found in mitochondria and endoplasmic reticulum of excitable cells. Its function is to remove the calcium ion from the cells. By doing this, sodium-calcium exchanger allowed sodium ions to flow down its concentration gradient across the plasma membrane and exchange for the counter transport of calcium ion. And this image may show you how a sodium-calcium exchanger works. As you can see in the image, 3 sodium ions binds to the transport carrier protein on its exterior side, and a single calcium ion bound to the same protein of the membrane interior side. Once both are bound, a conformational change occurs in a same protein channel which release energy. As a result, 3 sodium ions are transported to the interior side and a single calcium ion is transported to the exterior side. So, I think that's all from me. I will pass the following slides to the next presenter.
Why is cotransport so important? Well, cotransport is responsible for the accumulation of nutrients, neurotransmitters, ions, etc. from bacteria to man. This is of course vital for human survival as we need nutrients and such to become strong and healthy. Besides from that, cotransport is also important for mediating the movement of ions. This means that cotransporters ensure that ions are being transported to the other side of the membrane according to its specific method. So before we end this video, let me give you a brief summary of what was learned. Cotransport is the coupled movement of the first molecule going down its concentration gradient with the second molecule going against its concentration gradient. Cotransport is being carried out with the help of carrier proteins. A carrier protein is a specific binding site that is specific for its substrate. They transport solutes across the lipid bilayer by undergoing reversible conformational changes that alternately expose the solute binding site first on one side of the membrane and then on the other. There are two types of cotransports, which are sympod and antipod. Sympod is a process where a membrane protein facilitates the simultaneous transport of 2 distinct substances across the cell membrane in the same direction, whereas antipod is the process where a membrane protein transports the molecules at the same time in the opposite direction. And that is it from us, guys. Thank you for watching. I hope you enjoyed this video. Bye bye.